Hey guys, welcome to chapter 3 of the exam 1 for human physiology. And today I'm going to go over cell membranes, passive slash active transport, and resting membrane potential. Let's just get into it. The plasma membrane is a fluid lipid bilayer embedded with proteins, and the proteins are structured in a fluid mosaic model. More information into that in a bit. Phospholipids form a bilayer. The bilayer has a hydrophobic interior, which is sandwiched between a hydrophilic inner and outer surfaces. Now, what does that mean? Well, let's look into the structure of the phospholipid. As you can see, it has a hydrophilic head and a hydrophobic tail. And the way that it's structured is that the hydrophobic tail is facing towards each other, which makes the interior of the phospholipid hydrophobic and the outer hydrophilic, because remember, the head is hydrophilic. Um, this arrangement makes the phospholipid bilayer selectively permeable, meaning it allows some substances to pass through easily while the others are blocked. So some examples that can go through easily to the phospholipid bilayer is oxygen, carbon dioxide, steroids, or anything fat soluble. Now, some things that are blocked are glucose, proteins, charged molecules like sodium, potassium, calcium, and etc. all need the help of a channel to cross through. So, say sodium, for example, say sodium goes into contact with the phospholipid bilayer. What would happen is that the sodium would just bounce around. It'll go like this until eventually it'll go to the channel. Now, what are the channels? Well, channels are proteins. The proteins are embedded within the bilayer, creating a mosaic of molecules with various functions such as transportation, anchor, recognition markers, and etc. Now, if you look at the other proteins, they're not all the same, okay? Proteins in the phospholipid bilayer are all not the same. The proteins embedded in or attached to the phospholipid bilayer of the plasma membrane serve different functions and have different structures. Now going back into the channels, the channel basically serves as a transportation for impermeable molecules. So back with the sodium, it goes to the channel, right? Now, channels can be open or gated. Sorry. Now, open is basically self-explanatory. It's basically provides a steady continuous flow of ions, whereas gated, it can be open or closed, and it depends on the response to the specific signals. So, for example, mechanically, it opens in the response to physical forces like stretch or pressure. With voltage, it opens in response to changes in the electrical voltage across the membrane. And finally, chemically or ligand, it opens in response to when a specific molecule binds to the channel. Okay. Now, in the hydrophobic interior, there are also cholesterol molecules, which are tucked between the phospholipid molecules. What the cholesterol does right here is that it determines the fluidity of the membrane. It basically determines how much they can bend or flex. So if there is more cholesterol, then the phospholipid bilayer is more rigid, sorry, and the less cholesterol, which means it's less rigid. Carbohydrates are attached to its outer surface. So the carbohydrate chains are attached to the outer surface of the membranes, and it is often linked to the proteins or lipids. They act as markers or receptors. So the names play a big role into the name. Glyco, which means sugar, and protein, which means protein. So as you can see here, uh, sugar, protein, glycoprotein, and that's because it's attached to a protein. Sugar, fat, or glycolipid is because it's attached to the hydrophilic head or the fat. And so yeah, that's it with the carbohydrates. And then finally, its EM appearance is trilaminar. So what that basically means is that under an electron microscope, the plasma membrane appears trilaminar, meaning it has three layers: a dark line, a light space, and another dark line. This visual appearance is due to the arrangement of the hydrophilic heads and hydrophobic tails of the phospholipids. Okay. Um, the lipid bilayer has three functions. One, it forms the basic structure of the membrane. Again, basic structure. It forms the framework or the backbone of the cell membrane. Two, its hydrophobic interior is a barrier to water-soluble substances. So, barrier to water-soluble substances. The hydrophobic interior of the bilayer blocks most water-soluble molecules from passing through. Three, it allows the membrane to be fluid. So, fluidity. It helps maintain the flexibility of the membrane, allowing it to move and change shape. And so then we move on to the functions of membrane proteins. The membrane proteins have many functions. Some span the membrane as water-filled channels. Channels. Some proteins create channels that allow water and certain ions to pass through the membrane. Others serve as carrier molecules. Carrier proteins. These proteins transport substances across the membrane by binding them and changing shape. Some serve as docking marker acceptors. Docking markers, or some proteins, help other molecules dock at the membrane. like when signaling molecules to bind to cells. Membrane-bound enzymes are on the surface. Enzymes. Certain proteins act as enzymes that catalyze or speed up chemical reactions on the membrane surface. Receptor sites are proteins that bind with specific molecules. Receptors. Um, some proteins act as receptors that bind to specific molecules, triggering responses inside the cell. So, say hormones, for example. Some proteins are cell adhesion molecules. Adhesion molecules. These proteins help stick to one another and to structures outside the cell, such as the extracellular matrix, which we'll go over in a bit. Cells are held together by different means. Extracellular matrix serves as the biological glue. There's three kinds of protein fibers that are interwoven in this matrix. It is collagen, elastin, and fibronectin. Second one is desmosomes. They act as spot rivets to anchor adjacent cells that are not touching. So this is spot desmosomes. Um, Desmosomes are strong connection between the cells that acts like rivets, like I said on the slide, to anchor the cells that are not directly touching each other. This is important in tissues that need resistance, like the skin or heart tissues. Tight junctions. Cells adhere this way at points of direct contact. So in this case, tight junctions form a seal between the cells, ensuring that nothing leaks between them. They are important in places like the stomach or the intestines, where you would not typically want, or actually in any case, where you do not want any substances to leak between the cells. And finally, gap junctions. These small tunnels connect cells together. So in gap junctions, these are small tunnels between the cells that allow communication. They let small molecules and ions to pass between the cells, helping them work together, such as in the heart or in the nervous system. So an overall summary of these four is that for extracellular matrix, it's just think of it as something that holds cells like a glue. Okay. For desmosomes, it provides a strong connection for cells under stress. For tight junctions, it seals the cells to prevent leakage, and for gap junctions, it allows cells to communicate by passing small molecules. Okay. Intercellular communication occurs in three ways. One way is through gap junction. So again, I already went over gap junction, but I'll explain it more over here. So gap junction is basically um, a tunnel between cells. So the gap junction forms a small channel that connects the inside of one cell to the inside of another. So through these channels, small molecules, ions, and electrical signals can be passed back and forth between the connected cell. This allows to work. This allows the cell to work together quickly, sending signals almost, instantly. Some cells communicate by signal molecules on the surface membrane. So when we say that some cells communicate by signal molecules on the surface of a membrane, it means that one cell uses specific molecules on its surface to send a message to another cell. These molecules on the surface are recognized by receptors on a nearby cell, allowing the cells to communicate. So in a simple breakdown, one cell has a special molecule on its surface, basically like the keys. Okay, this is like the keys. The other cell has the receptors on its surface, basically like the locks. Um, the signal molecule from one cell fits into the receptor on this other cell, sending a message to that cell to do something, like to grow or move or to change. Okay. Then finally, we have extracellular chemical messengers, which is the most common means. There are four types, which are paracrines, neurotransmitters, hormones, and neurohormones. So with paracrines and neurotransmitters, it is local, whereas hormones and neurohormones, they are distant. So by local, it means that the signal affects nearby cells. And with distant, it means that the signals travel far away from where they are made, and they often have to travel through the bloodstream. Okay, so paracrines. Paracrines are local chemical messengers between the cell. Okay, so paracrine secretion or paracrine signaling is a type of cell communication where a cell releases signaling molecules that affect nearby cells. Basically, what I just said before. Unlike hormones that travel through the bloodstream to affect distant cells, paracrine signals only act locally, impacting cells in the immediate vicinity of the secreting cell. Um, in paracrine signaling, a cell sends signals to the other cells that are close by. This signal doesn't have to travel far, so it only affects the nearby cells and not the cells all over the body. Paracrine signaling is fast because the signal does not need to travel long distance. Before I continue on, I want to make a comment in regards to the cells. So for a cell to communicate, they must have receptors right here. These are the receptors. Because if a cell does not have a receptor, then what would happen is that the signal would just get bounced out. It won't be caught by the signal. Okay, let's continue. Um, Neurotransmitters are short-range chemical messengers released by neurons. So right here is a neurotransmitters. Neurotransmitters are chemical messengers that allow nerve cells or neurons to communicate with each other or with other types of cells, such as muscle cells. They transmit signals across a small gap between neurons called a synapse. This right here is a synapse, small gap. In a simpler breakdown, when a nerve cell or a neuron gets an electrical signal, it releases neurotransmitters from small sacs at the end of the neuron. Um, The neurotransmitter travel across the synapse, which is a tiny gap between between the two neurons or between a neuron and another type of cell. And on the other side of the synapse, on the other side, the neurotransmitters attach to the receptors on the next neuron or target cell. Once the neurotransmitter binds to the receptor, it causes a response in the receiving cell, such as sending a new electrical signal, releasing a chemical, or causing a muscle to contract. And that's for neurotransmitters. Hormones are chemical messengers secreted by the endocrine glands. So, hormonal secretions. Hormones are chemical messengers produced by the glands in the body that travel through the bloodstream to get to an organ or tissues where they trigger specific responses or regulate various bodily functions. Hormones play a key role in processes like growth, metabolism, reproduction, and mood regulation. So, how does the hormonal secretion work? Well, it is produced by glands. Hormones are made by specialized glands such as the thyroid, adrenal glands, or the pancreas. These glands are part of the endocrine system. Um, yeah. Uh, Once produced, the hormones are released into the bloodstream where they travel through the body to find their target cells. Each hormone binds to specific receptors on or inside target cells. These receptors are like locks, and the hormones is the keys that fit and triggers the response. So when a hormone binds to a receptor, it activates certain processes in the target cell. This could involve turning genes on or off, altering cell metabolism, or influencing the cell's behavior. Then finally, neurohormones, which are long-range chemical messengers secreted into the blood by neurosecretory cells. Um, right here. Neurohormones are chemical messengers that are produced and released by nerve cells or neurons, but act like hormones by traveling through the bloodstream to reach distant target cells. They combine features of both neurotransmitters, in which it is released by neurons, and hormones, in which it acts over a long distance through the blood. So, how do neurohormones work? Well, neurohormones are produced by neurons. Um, Neurohormones are made by specialized neurons, typically located in the parts of the brain like the hypothalamus. Unlike regular neurotransmitters, which act across small gaps or synapses, neurohormones are released into the bloodstream. So once in the blood, neurohormones travel throughout the body, reaching distant organs and tissues where they bind to specific receptors on target cells. When neurohormones bind to receptor cells on target cells, they cause specific changes, such as regulating hormone production, controlling metabolism, or influencing bodily function. And yeah, by signal transduction, incoming signals are conveyed to a target cell's interior for execution. A first messenger produces an intracellular response by opening or closing specific channels in the cell membrane to regulate ion movement, and then transferring the signal to an intracellular chemical messenger, or the second messenger. So, to put this into simpler terms, a signal transduction is when an outside signal, like a hormone or a neurotransmitter, triggers a response inside the cell. So what happens is the first messenger is the external signal molecule. It creates a intracellular response or a reaction inside the cell by opening or closing the channels in the cell membrane to control the movement of ions. This can change the cell's electrical state, which affects how the cell behaves. Um, After the first step, it then activates a second messenger inside the cell. The second messenger helps continue the signal inside the cell, leading to specific cellular response, like turning on a gene or making a protein, for example. So, in summary, the first messenger, like hormones, start the signal, while the second messenger helps carry the signal further inside the cell for action. Some extracellular chemical messengers open chemically gated channels. So this slide I basically already went over in the beginning of the video, but I'll go over it again. So leak channels are open all the time. So again, this is the channel. Channels can be open or gated, but the one that I just read is open or leaked. So open or leak, they are always open. Ions can flow freely through these channels all the time without needing any specific signal or trigger. Gate channels can be open or closed in response to various controlling stimuli. There are three kinds of gated channels. Some bind to chemical messengers. Chemically, these open or close when a specific chemical, like a neurotransmitter, binds to them. Some respond to electrical changes, voltage or electrical. These respond to changes in electrical charge across the membrane, like during nerve impulses. Finally, some are sensitive to stretching/mechanical changes. These are open or closed in response to physical stretching or changes in the membrane shape. So in summary, leak channels are always open, while gated channels require a signal to be opened or closed. Two secondary messengers are cyclic AMP and calcium ions. So what this whole slide is talking about are the secondary messengers, which are the molecules that help transmit signals inside a cell. And the two examples that are provided are the cyclic AMP and calcium ions. It says here, secondary messengers initiate a similar cascade of intracellular event. What does that mean? Well, the secondary messengers starts a series of internal processes inside the cell after the initial signal or the first messenger is received. Okay, oops. These events change the shape and functions of particular proteins. So these processes can change in specific proteins inside the cell, altering their shape and function. That's basically what it means. These changes cause cellular responses. What does that mean? Well, these proteins changes lead to different responses from the cell, such as activation or deactivation of certain activities. The steps of secondary messenger pathways amplify the initial signal. Uh, what is that saying? Well, the steps in these pathways amplify the original signal, meaning that a small signal can cause a large response inside the cell. So in short, secondary messengers help ensure the signals from outside the cell to cause the right reactions inside the cell. Okay. An example of secondary messenger system, CAMP. So this image right here, this image illustrates a signal transduction using the example of how a hormone, or the first messenger, triggers a cascade of events inside a cell via secondary messenger like the cyclic AMP. So here's the step-by-step breakdown of what's going on. So first, we have a hormone, or the yellow right here, as the first messenger, which then it binds to a specific receptor on the cell membrane, which is this purple right here. Um, this activates the receptor. Um, the receptor then activates an attached G protein, or the purple relay molecule, and the G protein is switched from its inactive form, which is bound to GDP, to activate form, to bound to GTP. The activated G protein moves along the membrane and activates an effector enzyme, which is the adenylate cyclase. This enzyme then starts producing the secondary messenger. The effector enzyme converts ATP, which is adenosine triphosphate, into cyclic AMP, which is cyclic adenosine monophosphate, which is the secondary messenger. So ATP and CAMP, what ATP is adenosine triphosphate, tri meaning three, so there's three phosphates, whereas cyclic AMP, which is cyclic adenosine monophosphate, which basically means mono equals one, it's saying that this is implying that the ATP loses two phosphates, making it into a monophosphate. Okay. Okay. Then the effector enzyme converts ATP into cyclic AMP, which is the secondary messenger. I already went over that. The CAMP, or the cyclic adenosine monophosphate, then activates a protein kinase, or however you say that, inside the cell, which are the enzymes that trigger various cellular responses. And these responses include activating other enzymes, stimulating the release of substances like hormones or neurotransmitters, and opening opening ion channels to allow ions to move in and out of the cell. So in summary of this whole image, this pathway shows how a hormone triggers a cascade of reaction inside a cell, amplifying the signal through secondary messenger to produce specific cellular responses, such as activating enzymes or opening channels. This is an example of how external signals are translated into internal actions within the cell. Membrane transport can be unassisted. Diffusion is one example. Diffusion depends on the random movement of molecules. By net diffusion, molecules move down their concentration gradient. Movement is always from areas of high concentration to areas of low concentration. Net diffusion tends towards a steady state. Diffusion can occur through a permeable membrane, and substances can pass through a lipid bilayer or channel. So in a simple breakdown, what it's saying is that diffusion, by definition, refers to the movement of molecules from areas where they are high in concentration to areas where they are low in concentration. This happens naturally due to the random movement of molecules. Then, concentration gradient. What does that mean? Well, diffusion occurs because molecules want to spread out evenly. So if there are more molecules in one side of the membrane, they will move toward the side with fewer molecules. Net diffusion. So there is a difference. Well, there isn't much difference between diffusion versus net diffusion. Again, diffusion refers to the overall process of molecules moving from an area of higher concentration to an area of lower concentration, whereas net diffusion refers to the overall or net movement of molecules in one direction. So even though molecules are moving randomly in all direction, net diffusion focuses on the overall movement from an area of high concentration to area of lower concentration until the equilibrium is reached. So in summary, diffusion is the random movement of molecules, while net diffusion emphasizes the overall direction movement during the diffusion process. Um, so here, let's look at the image in the bottom. So the diagram or the image in the left shows when the membrane is permeable. So the molecules move from left, which is the higher concentration, to the right, to the lower concentration until equilibrium is reached. Whereas in the image in the right shows the membrane is impermeable. So the molecules stay on the left side, being unable to cross the membrane. So this image right here represents a simple diffusion. So the key points on what we have to know about simple diffusion is that molecules move from area of higher concentration to areas of lower concentration down the concentration gradient. So as you can see, look at these yellow dots or the lipid soluble solutes. It's moving from an area of higher concentration to areas of lower concentration. And that's because it's trying to reach equilibrium. And you can say the same with the red dot. There's more red dots in the extracellular fluid, so it's trying to go to the cytoplasm to equilibrate. Okay. Um, the lipid soluble solutes, okay, sorry, these yellow dots right here, or these ones, these are molecules that can dissolve in lipid fats, like small nonpolar molecules. These molecules can pass through directly the lipid bilayer without needing any help, as shown by the yellow molecules passing through the bilayer. So like this, it's just going through without needing any assistance. However, if you look at the small lipid insoluble solutes, like these ones, these molecules, like small ions or polar molecules, cannot dissolve in the lipid bilayer because they are not fat soluble. These molecules represented by the small red dots need help to cross the membrane, so they use a channel protein. This is right here is a channel protein. The channel proteins are embedded in the lipid bilayer that forms channels for small lipid insoluble solutes to pass through. In this case, the red solute is moving through a channel protein because it cannot pass through the lipid bilayer on its own. Um, and so yeah, there isn't much to say about this other than, you know, molecules move down from, move down from areas of higher concentration to areas of low concentration, and lipid soluble molecules moving directly through the bilayer, while lipid insoluble molecules need protein channels to cross the membrane. Activities in the human body where diffusion occurs include the exchange of oxygen and carbon dioxide between the blood and the air in the lungs, and the movement of substances across kidney tubules. The diffusion of sodium and potassium ions through open channels in the plasma membrane. So in this slide, what is that saying? Well, it's saying that sodium and potassium ions move through open channels in the cell membrane, and it is driven by their concentration differences. This movement is crucial for processes like nerve signaling and muscle contraction, where the flow of these ions help create electrical impulses. By Fick's law of diffusion, 5 factors affect the rate of diffusion. One is the magnitude of the concentration gradient. So the larger the difference in the concentration between two areas, the faster the rate of diffusion. Two, the permeability of the plasma membrane to a substance. So if the membrane allows the substance to pass through easily, like with higher permeability, diffusion occurs more rapidly. Three, the surface area of the membrane across which diffusion takes place. A larger membrane surface area provides a more space for diffusion, leading to an increased rate of diffusion. Four, the molecular weight of a substance. So smaller molecules diffuse faster than larger ones. The lower the molecule weight, the quicker the diffusion. Finally, five, the distance through which diffusion takes place. So the shorter the distance the molecules need to travel, the faster the rate of diffusion. So in simpler terms, the bigger the difference, the faster the diffusion. If it's easier to pass, the faster the diffusion. If there's more space, the faster diffusion. So what does that mean? Well, if there's more surface area on the membrane, it can spread out faster. Smaller things move faster, and shorter distance equals faster diffusion. Okay. Ions that can permeate the membrane also passively move along their electrical gradient, opposites attract. A difference in charge between two adjacent areas produces an electrical gradient. This is called a potential. the net effect of electrical and concentration gradient produces an electrochemical gradient. Now, you might be confused as to what I just read, so I'm just going to put a video here explaining it.
I'm going to talk now about electrochemical gradients, which are a very important concept when it comes to the physiology of all cells. People often associate electrochemical gradients just with nerve cells, but I'm going to explain that in fact electrochemical gradients are important for the functioning of any cell in the body, nerve cells representing a special case which can use the energy in these gradients to power action potentials. We'll come to that a little bit later. But to begin with, I'm going to start with a very hypothetical situation. I'm going to imagine that we have a container, and the container is divided into two parts, and there's a membrane in the middle of the container dividing two solutions. On the left-hand side, we'll have a concentrated solution of glucose. Let's say that it's 100 millimoles per litre glucose solution. On the right-hand side, let's have a more dilute glucose solution. It's 10 millimoles per litre. Now, the membrane down the middle is going to be permeable only to glucose. We're going to say that it's not permeable to water at all. So this is not an osmosis experiment. We're not interested in water moving across the membrane. We're only interested in the movement of glucose. And the question is, what's the glucose going to do? Now, clearly, there's a concentration gradient for glucose, which is going from left to right. And so glucose is going to tend to want to cross the membrane into the right-hand compartment, which has the dilute solution. Now, this will carry on until eventually the concentration of glucose on both sides of the membrane is exactly equal. It's the same. Now, that's not a very interesting situation, but I want to contrast that to what you would see in a very similar experiment if instead of glucose in the two compartments, you had potassium chloride. So now on the left-hand side, we've got 100 millimoles per litre of potassium chloride. And that means you've got potassium ions, which are positively charged, and chloride ions, which are anions, they're negatively charged. And they're all floating around in the solution. Now, in any solution of potassium chloride, you're going to have the same number of positive ions and negative ions. And so there's no charge imbalance, if you like. There's no extra positives, there's no extra negatives. It's a neutral solution, and it contains both types of ion. Now, this is a relatively concentrated potassium chloride solution, and I'm going to say that this side of our little experimental cell, this is like the inside of a real cell, because the inside of a real cell has relatively concentrated potassium within it. Now, the right-hand side of our little experimental cell is going to have a dilute potassium chloride solution, 10 millimoles per litre, we'll say, and also, to begin with, that has an equal number of positives and negatives, and so there's no charge imbalance. It's a neutral solution once again. There are just fewer potassiums and chlorides than there are on the left side. And this is going to be analogous to the extracellular fluid, which is more dilute in terms of potassium than what's inside a cell. Rather like with the glucose experiment, we've got a concentrated solution on the left and a more dilute solution on the right. But in this case, I'm going to say that the membrane separating the two sides is permeable only to potassium. Now, that's going to be relatively similar to a real cell membrane, because as we'll see, real cell membranes are permeable mainly to potassium ions, more so than to some of the other ions that you might find in the cell. Now, the reason that a cell membrane might be permeable mainly to potassium ions is that it has a high proportion of potassium channels within the membrane. And those are going to be protein molecules which allow potassium through, but they don't allow anything else. So we have a situation now with a concentrated solution on the left-hand side, that's the interior of our cell, a more dilute solution on the right-hand side, that's the extracellular fluid, and a membrane separating the two, which rather like a real cell membrane, is mainly permeable to potassium ions. In this case, we'll say only permeable to potassium ions. What's going to happen? Well, we have a concentration gradient for potassium and for chloride, but chloride can't cross the membrane, so it's stuck where it is. Potassium can cross the membrane, and because there's a concentration gradient from left to right, some potassium will move into the right-hand compartment. So the concentration gradient drives potassium into the right-hand side. But this is where we have a situation different from the one that we saw with the glucose earlier, because potassium has a charge. It's a cation, it has a positive charge. And so for every potassium that moves across the membrane, without chloride following, because it can't, we have an extra positive charge on the right-hand side, which we didn't have before. And of course, for every potassium moving through to the right, that's going to leave behind a negatively charged chloride on the left, which is now unpaired. So what's going to happen is that as more and more potassium crosses the membrane, the right-hand side becomes more positive, the left-hand side becomes more negative. And so we're creating an electrical gradient across the membrane. Now, if you're a potassium ion, you're now experiencing two forces. You have the concentration force from left to right, but you also now have an electrical force, which is going from right to left, because you're a positively charged ion. And from the point of view of your charge, you would rather be on the left-hand side. So unlike for glucose, you now have two forces in opposite directions, a concentration force left to right, an electrical force right to left, and what's going to happen is that exactly the right number of potassium ions will move across the membrane, such that the developing electrical force will become equal and opposite to the concentration-based force, and at that point, there'll be no more tendency for potassium to move. You then have an equilibrium situation. The forces are equal and opposite, and at that point, there's no more net movement of potassium ions. So what we have at this point is a situation where potassium can still move to the right, or it can move to the left, but there's no more net tendency for it to move. Potassium is at equilibrium. But at that equilibrium position, we have a membrane which now has a potential difference across it, an electrical difference across it. Inside, the inside of the cell is negative, outside, the extracellular fluid is positive. So in principle, we could put a voltmeter across the membrane, we could measure that potential difference, and that would be a voltage. Now, you could calculate the value of that voltage if you wanted to. There's an equation which would allow you to do it, but we won't go into that at the moment. It's sufficient to say that if you have a membrane permeable only to potassium, and you have two different concentrations of potassium on one side and the other, potassium will move until there's a voltage across the membrane. That voltage technically is called the equilibrium potential for potassium. Now that, in fact, is very similar to what we would find if we looked at a real cell, because, as I mentioned earlier, a real cell has relatively concentrated potassium inside, relatively dilute outside, and the membrane is permeable to potassium ions. So some potassium would leave the cell across the membrane, bringing a positive charge with it, and that would leave the membrane with an electrical potential difference across it. Now, in the case of a real cell, that potential difference is called the resting membrane potential of the cell. And in a real nerve cell, the resting membrane potential takes a value of minus 70 millivolts. It's always presented as the inside relative to the outside. So minus 70 because the interior of the cell is more negative than the extracellular fluid on the outside. Now, it turns out that in a real cell, the situation is a bit more complicated than what I've just described. And that's because there are lots of ions which can cross the membrane, not just potassium. And notably, sodium complicates things because sodium can move across the membrane too. So in a real cell, the resting membrane potential is not exactly equal to the equilibrium potential for potassium, but it's very close. And if you can understand what potassium is doing, then you're pretty close to understanding what a real resting cell is doing and why we have a membrane potential across that membrane.
So what you need to take away from the video is in regards to the concentration gradient, electrical gradient, and the electrochemical gradient. So again, concentration gradient refers to the difference in the number of ions or molecules across the membrane. Ions naturally want to move from areas of higher concentration to areas of lower concentration. This is called diffusion, and it occurs without the need for energy. Electrical gradient is when ions move because of the difference in charge between two areas. So opposites attract. So positive ions move towards negatively charged areas, and negative ions move towards positively charged areas. And then you have electrochemical gradient. This is the combination of both electrical gradient and the concentration gradient. Ions move in a direction that balances both gradients. This means that ions move not only because of the charge differences, but also due to the difference in their concentration on either side of the membrane. So when the slide says a difference in charge between two adjacent areas produces an electrical gradient called potential, this refers to the separation of electrical charges across a distance, creating a difference in electrical energy between two regions. So let me break it down. In biological systems, different ions like, I don't know, sodium, potassium chloride are distributed unevenly across the cell membrane. Um, for example, the inside of a cell may have more negative charges while the outside has more positive charges. These two areas could be the inside and the outside of a cell. Because there are more positive ions outside and more negative inside, this sets up a charge difference across the membrane. The difference in electrical charges in regards to the positive versus negative between two regions creates an electrical gradient. This gradient is like a force that drives ions to move from one area to another. So positively charged ions are attracted to negatively charged areas, and negatively charged ions are attracted to positively charged areas. And so the electrical potential or the membrane potential is the measure of the energy difference between two areas due to the separation of charges, and it is often referred to in millivolts. So for example, the resting membrane potential of a neuron is typically negative 70 millivolts, meaning that the inside of a neuron is 70 millivolts more negative than the outside. So in short, the electrical gradient is a result of the different charges between two areas, and the potential is the energy stored because of the charge difference. This is what cells use to create and transmit signals. And then so the net effect of electrical and concentration gradient produces an electrochemical gradient. So in this diagram right here, let me explain what's going on. So in here, it's saying that the positively charged area moves to the negatively charged area because opposites attract. So these positive charges would go this way. And it'll go here like this, like this, like this, like this, like this. And these ones disappear because they're going this way, remember. And then eventually, the negatively charged area would reach an amount of positively charged area in which if a positively charged ion or cation would go this way, it would get pushed back because of the positively charged area in this place. And once that happens, this is called the equilibrium potential. Osmosis is the net diffusion of water down its own gradient. Now, reading this might be confusing, so I think it's better to just explain it in the picture. So, what is going on in the picture? Well, the left and right compartments contain solutions with solute, which is the red dot, and the solvent, which is just the water. Okay, the membrane between the compartments only allow water to move and not solute particles. So this membrane right here only allows water to move. So even though both oops sorry even though both compartments have the same osmolarity, which basically means concentration of solute, the right compartment has larger volume of solution because water can move to balance the concentration. So the water basically flowed from left to right to equalize the concentration, resulting in more water in the right compartment. This basically demonstrate how osmosis seeks to balance solute concentration by moving water, even if the solutes themselves can't pass through the membrane. Okay. Tonicity of a solution is the effect of the solution has on a cell volume. So in an isotonic solution, it basically means it has the same concentration of non-penetrating solutes as normal body cells. So in other words, the concentration of solutes is the same inside the cell as it is outside the cell. And you could also say the same with the water. Water moves in and out of the cell equally, so the cell's volume stays the same. A hypotonic solution has a lower concentration of non-penetrating solutes compared to normal body cells. Again, in other words, the concentration of solutes outside the cell is lower than inside the cell. So water moves into the cell, causing it to swell and possibly burst. A hypertonic solution has a higher concentration of non-penetrating solutes compared to normal body cells. So the concentration of solutes outside the cell is higher than inside the cell. So water moves out the cell, causing it to shrink. So in short, tonicity basically refers to how different concentration of solutes in a solution is and can cause cells to either shrink, swell, or stay the same. And it depends on the movement of the water. So in shorter terms, what it's basically saying is that for isotonic, it's balanced and no change is made. With hypotonic, water goes in, the cell becomes bigger. And hypertonic is when the water goes out and the cell shrinks. And yeah, okay. Membrane transport can be assisted. Carrier-mediated transport occurs by a carrier molecule flipping and changing its shape. So a carrier-mediated transport means that a molecule, like a glucose or ions, needs help from carrier proteins to cross the membrane. This carrier protein changes its shape to move the molecule. It can be active or passive. Active meaning it uses energy, like adenosine triphosphate, and passive meaning no energy is used. It has three important characteristics. Specificity, in which it means that the carrier only helps certain molecules, like a key to fit only one lock. Saturation, in which there's a limit to how many molecules can be carried at one time. And competition, in which if two types of molecules can use the same carrier, they basically compete for it. Facilitated diffusion is a type of assisted transport. By this process, a substance moves from a higher to lower concentration. So it's like basically regular diffusion. Um, Unlike simple diffusion, however, facilitated diffusion requires a carrier molecule. Glucose is transported into the cells by facilitated transport. So in simpler terms, it's basically saying that substances move from areas of high concentration to low areas of concentration, just like diffusion. But facilitated diffusion needs a helper protein because molecules like glucose are too large or they're not soluble in lipids to pass through the membrane on their own. So it's using glucose as an example in which the glucose enters the cell through facilitated diffusion using a specific carrier protein. And I want to note that this whole process is passive, which basically means that facilitated diffusion does not require any energy. Okay. A active transport is a type of assisted transport. Um, a substance moves against its own gradient, and it requires a carrier molecule. So it's basically saying that a substance moves against its own gradient, meaning it goes from a low concentration to a high concentration. And this process requires ATP because it's moving against its own gradient. It's like moving uphill, and it uses a carrier molecule, which helps the substance pass through the membrane. Primary active transport requires the direct use of ATP to drive sodium-potassium pump. The secondary active transport is driven by a concentration a gradient of ions established by primary active transport. So what primary active transport is saying is that it directly uses ATP to move the things, whereas secondary transport is using the leftover energy from the primary transport to move over things. So as I've previously said, this right here, say this is the primary active transport. It's using the direct use of ATP to move. And then right here is the secondary active transport, and it uses the leftover energy for primary transport to move move over things. Okay. Um, Other kinds of assisted transports are vesicular transport. in which the materials move in or out of the cell wrapped in a membrane. Examples of vesicular transport is endocytosis and exocytosis. By endocytosis, substance move into the cell, whereas exocytosis is the reverse process in which it is out of the cell. Um, to make sure that you guys don't, confuse between endo and exocytosis, just look at the first lettering of the word. So exo literally meaning out, cyto means cell, cyst means process of. So overall, exocytosis means the process of moving things out the cell. And it's the same said with endocytosis. Endo meaning in, cyto meaning cell, cyst meaning process of. And overall, it means process of moving things in the cell. So if you look at the image right here that is provided, what's happening here is exocytosis. Exocytosis. And how do I know that? Well, the image shows that it's moving out of the cell. So as you can see here, it is wrapped in the membrane. Oops. The cell is wrapped in a membrane where it goes to the phospholipid bilayer to be released, like that. Okay. Oh, okay. And now the rest over here are all endocytosis. Again, endo meaning in, cyto means cell, sys meaning process of moving things into the cell. So these three images are all endocytosis. Now, phagocytosis is also known as cell eating. Okay, phagocytosis is a process where the cell engulfs large particles like bacteria, dead cells, or debris. But in this example, for example, it's using food particle. What happens then is that the cell membrane extends around the particle, forming a vesicle called a phagosome, which then merges with a lysosome to break down the material. So lysosome comes and helps break down the food particle. Next up, we have bulk phase endocytosis, also known as pinocytosis or cell drinking. So with the bulk phase endocytosis, it involves the nonspecific uptakes of fluids and dissolved substances from the surrounding environment. The cell invaginates or folds inward, in which forms a vesicle and brings in extracellular fluid along with the dissolved molecules. And so yeah, that's a bulk phase endocytosis. And finally, we have receptor-mediated endocytosis. What that is a more specific process where the cell takes in particular molecules after they bind to receptors on the cell surface. The binding triggers the formation of a vesicle, which brings the bound molecules like hormones or nutrients inside the cell. Um, so, in quick summary, phagocytosis is for engulfing large particle or debris. Bulk phase endocytosis or pinocytosis is for taking in fluids and dissolved substances non-specifically. And receptor-mediated endocytosis is for selectively taking in specific molecules via receptors. Okay. All cells establish a membrane potential. This is a separation of charges across the plasma membrane. This potential develops due to differences in the concentration and the permeability of key ions. A cell develops a resting membrane potential when it is not sending electrical signals. So basically what this slide is explaining is the concept of membrane potential, which is the separation of electrical charges across a cell's plasma membrane. So here's a breakdown. Um, what a membrane potential is when all cells maintain a difference in electrical charge across their plasma membrane. This charge difference creates an electrical potential. So why does it happen? Well, this potential is caused by the differences in ion concentration and differences in ion permeability. So in regards to differences in the ion concentration, ions like sodium and potassium are not equally distributed inside and outside the cell. Typically, there's more potassium inside and more sodium outside. Oops. Okay. And in regards to the differences in ion permeability, this is basically saying that the membrane is more permeable to certain ions like potassium than others, which means that potassium can move more easily across the membrane than sodium, contributing to the charge differences. So, resting membrane potential. When the cell is not sending any signals, it is in its resting state. It has a stable charge difference across its membrane. This is called the resting membrane potential, and it's usually about negative 70 millivolts inside the cell, meaning that the inside of the cell is more negatively charged compared to the outside. So, in summary, membrane potential is the result of the uneven distribution and movement of ions across the cell membranes, leading to a change in to the difference when the cell is at rest. Okay. Sodium and Oops, sorry. I mean, potassium and sodium are critical. So the image right here that is, in the picture, emphasizes the importance of potassium ions, which is K+, and sodium ions, which is Na+, in establishing and maintaining the resting membrane potential across a cell's plasma membrane. So here's a simplified explanation. Sodium, or the Na, is primarily found in higher concentrations outside the cell, whereas potassium, or K+, is found higher concentration in the cell, so it's inside the cell. So the difference in concentration of these ions on either side of the membrane creates an electrochemical gradient. The sodium-potassium pump helps maintain this gradient by moving sodium out of the cell and potassium into the cell. This movement of ions is essential for many cellular functions. So the unequal distribution of these charged ions contribute to the negative charge inside the cell, relative to the outside, which is the resting membrane potential. So in a brief summary of what I just said, sodium or Na plus is mostly found outside the cell, and potassium is mostly found inside the cell. Um, the cell membrane uses something called a sodium-potassium pump to keep this balance. It does this by pushing sodium out and bringing potassium in. This creates a small electrical charge. The sodium pump has a small effect on the membrane potential. It transports more sodium ions to the ECF than potassium ions to the ICF. By this pump, more sodium is in the ECF and more potassium is in the ICF. So what that basically means is that the sodium potassium pump is actively transporting more sodium ions out of the cell to the extracellular fluid and more potassium ions to the into the cell, which is the intracellular fluid. This maintains a high concentration of sodium outside the cell and potassium inside the cell. A resting membrane is more permeable to potassium. This means that potassium can move freely in and out of the cell, particularly out of the cell into the ECF, the extracellular fluid. Therefore, more potassium diffuses into the ECF compared to the diffusion of sodium into the ICF. That's basically saying since more potassium can diffuse out than sodium can diffuse in, potassium contributes more to the resting membrane potential. The potassium equilibrium potential has a major effect on the plasma membrane at rest. That basically means that the potassium equilibrium, the potassium equilibrium potential is at negative 90 millivolts, has a greater influence on the overall membrane potential of the cell. The resting membrane potential is often around negative 70 millivolts, so potassium's movement is a key factor. So simply put, the sodium-potassium pump helps maintain a balance, but potassium's ability to move across the membrane more easily has a larger effect on the resting potential of the cell. Okay. And so let's look at the image here. This image explains how potassium ions and the gradient affects the membrane potential, specifically focusing on the equilibrium potential for potassium. All right, the key points. We look at the concentration gradient for potassium. So inside the cell, or the ICF, there's a high concentration of potassium, whereas the outside of the cell, the ECF, there is a lower concentration of potassium. This concentration gradient drives potassium to move out the cell from high to low concentration. Then we look at the electrical gradient for potassium. So as potassium ion leave the cell, the inside of the cell becomes more negatively charged. This creates an electrical gradient that attracts potassium back into the cell since opposites attract. Um, and then we look at the equilibrium potential. So the equilibrium potential for potassium is around negative 90 millivolts. This at the voltage at which the electrical gradient in which it's pulling the potassium back in balances the concentration gradient pushing the potassium out. The plasma membrane. So this barrier controls the movement of ions between the ICF and the ECF, helping to maintain the electrical gradient. So, to put it simply, the movement of potassium would stop when the concentration gradient leaving is equal to the electrical gradient pushing it back in, and that is around negative 90 millivolts. Um, in simple terms, potassium wants to move out of the cell due to its high concentration inside, but negative charge inside the cell pulls it back. Then, when these two forces balance out, the membrane potential stabilizes around negative 90 millivolts for potassium. The effect of concurrent potassium and sodium movement establishes the resting membrane potential. The distribution of chloride ions is passively driven by the established membrane potential. These ions concentrated in the ECF. Okay, so going over this image shows a resting membrane potential. And it is basically the electrical difference across a cell's plasma membrane when the cell is not actively sending a signal. And when it is not sending a signal, it is measured at about negative 70 millivolts, meaning that the inside of the cell is more negatively charged than the outside of the cell. Okay, so the key players that we're going to be looking at are potassium, the anion right here, sodium, and chloride. Starting off with potassium, potassium is more concentrated inside the cell compared to the outside. So potassium tends to leak out of the cell because of its concentration. It diffuses from high to low concentration to the outside. However, as potassium leaves the cell, it makes the inside cell more negative. The negative charge inside eventually pulls the potassium back to the cell. Um, this is because same charges repel and opposites attract. So when potassium leaves the cell, its positive charge gets stuck outside of the cell, in which it is attracted to the negatively charged area inside the cell. This balance between potassium leaving due to its concentration and being pulled back in due to the negative charge is part of what maintains the resting memory potential. Okay. Next up, we have the large anion right here. These are negatively charged molecules like proteins that are too large to leave the cell. So they just stay inside to contribute to the negative charge inside the cell. For chloride, chloride is more concentrated outside the cell, but is generally repelled from entering the cell because of the negatively charged inside. This means that chloride movement doesn't significantly affect the resting memory potential. Then we look at sodium. Sodium is more concentrated outside the cell. Sorry. Although sodium wants to enter the cell, the membrane is not very permeable to sodium, so only a little sodium enters, which would make the inside more positive. Although it tries to make the inside more positive, the best it can do is give plus 20 millivolts, and it'll still be at negative 70 millivolts, which is still the resting membrane potential. Okay. Okay. Operation of the sodium-potassium pump. So going over each step, we first have sodium binding, which first starts here. Okay. In sodium binding, what happens is three sodium ions from inside the cell bind to specific sites on the pump proteins. This triggers the pump to begin its function. Okay, second step is phosphorylation. The pump is phosphorylated. This means that a phosphate group from the ATP attaches to the pump, causing it to change its shape. ATP is used to provide energy for this process, leaving ADP behind. Third step is shape change and sodium release. So the shape change of the pump protein causes three sodium ions to be released outside of the cell into the extracellular fluid, ECF. Then in the fourth step, the potassium, well, two potassium ions from outside the cell then binds to the pump protein, triggering another change in the pump shape. Fifth is phosphate release. The binding of potassium triggers the release of phosphate group, which had earlier attached from ATP. This restores the pump's original shape. And then finally, six, potassium release and sodium sites ready. The restored pump shapes moves the two potassium ions inside the cell. At this point, the pump is ready to bind to more sodium ions and the cycle repeats. Okay. Nerve and muscle cells can use the membrane potential to send electrical signals between the cells. So this image right here illustrates the action potential cycle in neurons, which is how nerve cells send electrical signals. So here's a breakdown of the phases in simpler terms. We first have the resting state. The resting state is around negative 70 millivolts. So at rest, the neuron is about negative 70 millivolts, as stated, which is the resting membrane potential. Sodium and potassium channels are closed, maintaining the stable state. We then get into the depolarization phase. When neuron is stimulated and reaches the threshold, sodium channels open. Sodium ions flood into the cell, making the inside of the cell more positive, which is up to 30 plus 30 millivolts right here. And then we get into the repolarization phase. After the depolarization, sodium channels close and potassium channels open. Potassium ions move out of the cell, returning the inside of the cell to a negative charge. Okay, and then the fourth stage, which is called the undershoot or the hyperpolarization. What happened here is that the potassium channels remain open a little too long, making the cell more negative than its resting potential. This is called the undershoot. Okay, so and then after that, it is the return to the resting potential. The potassium channels eventually close and the cell returns to its resting membrane potential of negative 70 millivolts, which is again ready for another action potential. So some key points to take away from this image is the spike or the action potential. This occurs due to the rapid influx of sodium, which makes the cell more positive. Another key point you should take away from this is the return of negative states that happens as potassium ions leave the cell. And that should be it with chapter 3 of the exam 1 for human physiology.